Hi, welcome back to Psychology 201 with your instructor, Trevor Setman. So today, what we're going to be going over is we're going to look at the biological bases of behavior. So essentially, we've now gone through research methodology and approaches to psychological research and kind of the background of psychology in the very first chapter. And now we're going to kind of go into looking at the biological background of psychology and basically how the brain functions, what are the different elements. So just a snapshot of the chapter. First, we're going to be going over communication in the nervous system, looking at organization of the nervous system, research methods, the brain and behavior, cerebral laterality, the endocrine system, and heredity and behavior. So first, we start with the fundamental base of how the brain communicates. Okay, so in this chapter, it starts out by beginning with communication in the nervous system. And before we can really delve into that, we need to kind of know what the hardware is. What are the different parts of that specific process, and how do they function as a whole? So first, I've listed the different parts. There's also a diagram here. First, we have the glia, which is the structural support and insulation. Neurons, which are the communication bodies. The soma, which is the cell body. Dendrites, which receive the information. Axon, which transmit the message. Terminal buttons, which secrete the neurotransmitters. And the synapse, which is junction where information is transmitted. And essentially, neurons are the communication links of the nervous system. It's how it communicates. The diagram highlights the key parts of the neuron, including the specialized receptor areas known as the dendrites, the cell body, which is the soma, the axon fiber along which impulses are transmitted, and the terminal buttons, which release chemical messengers that carry signals to other neurons. So neurons vary considerably in size and shape, so they're usually densely interconnected. So that's kind of just a basic overview of the communication happening and the process that underlies that communication in the brain. So next we move on to look at neural impulses. So first, the chapter talked about Hodgkin and Huxley's several studies looking at the cells of giant squid, and basically looked at the fluids inside and outside the neuron, the electrically charged particles, the ions, and neuron at rest, negative charge on inside compared to the outside. And the electrochemical properties of the neuron allow it to transmit signals. So in other words, think of it as a message system. The electric charge of the neuron can be measured with a pair of electrodes connected to a oscilloscope, as Hodgkin and Huxley showed with their squid axon. Because of its exceptionally thick axons, the squid has frequently been used by scientists studying the brain and studying the different components that are communicating within that specific system. A couple things to note. At rest, the neuron's voltage hovers around negative 70 millivolts. So that's what's called the resting potential. Now we move on to look at the action potential. And this is essentially the stimulation which causes cell membranes to open briefly and accept those different chemical communications that are occurring due to the neurons. They're positively charged sodium, ions flow in, and then there's a very brief shift in electrical charge that travels along the axon. When stimulated, there's a brief jump in a neuron's voltage, resulting in a spike. And then when we're looking at it, we do see electrical activity. Some of you may have not thought of the brain as having electrical components, but really there are electrical processes going on in the brain. The change in voltage, as we discussed, is called the action potential, which travels along the axon like a spark traveling along a trail of gunpowder. So here we move on to look at synapses. And basically, as the diagram shows, when a neural impulse reaches an axon's terminal buttons, it triggers the release of chemical messengers called neurotransmitters. Okay, these are what are transmitting the neurons. The neuron transmitter molecules diffuse across the synaptic cleft and bind to receptor sites on the postsynaptic neuron. A specific neurotransmitter can bind only to receptor sites that its molecular structure will fit into, much like a key would fit into a lock. So those need to be adapted for that specific function. In other words, if it won't fit, those neuron transmitters aren't able to access it. So a couple things to discuss, things to know for this chapter is looking at the synaptic cleft, which we'll be talking about in a little bit, and we have a video clip looking at that as well. The presynaptic neuron, synaptic vesicles, neurotransmitters, and then postsynaptic neuron and the receptor sites. So that's briefly what we just talked about. And this diagram really illustrates some of those components and how they really relate to each other. So instead of just memorizing definitions, you're going to want to focus on some of these diagrams as you go through the chapter and kind of see how these are all interrelated and connected. So looking at neural impulses, the main elements in synaptic transmission are summarized in this clip. So we're going to watch this really briefly and then we'll come back.
When an action potential reaches the synaptic terminal of the axon, it encounters a region called a synapse where two neurons come close together but do not touch one another. A minuscule gap, called the synaptic cleft, separates the synaptic terminal of the first or presynaptic neuron from the dendrite or cell body of the second or postsynaptic neuron. The arrival of an action potential at the synaptic terminal causes the synaptic terminal to become positively charged and release neurotransmitters into the synaptic cleft. The neurotransmitters diffuse rapidly across the synaptic cleft and bind to receptors in the membrane of the dendrites or cell body of the postsynaptic cell. Each type of receptor in the postsynaptic membrane binds to a specific type of neurotransmitter, and after binding to a neurotransmitter, the receptor causes specific types of ion channels in the postsynaptic membrane to open. As ion channels are opened, ions flow across the cell membrane of the postsynaptic neuron along their concentration gradients. This flow of ions into the postsynaptic neuron causes a postsynaptic potential in the dendrites or cell body. Depending on the channels open and the ions that flow in, synaptic potentials can be either excitatory, making a neuron less negative inside and more likely to fire an action potential, or inhibitory, making it more negative and less likely to fire. An excitatory postsynaptic potential is called an EPSP. An inhibitory postsynaptic potential is called an IPSP. A synapse that produces EPSPs in the postsynaptic cell is called an excitatory synapse, while a synapse producing IPSPs is an inhibitory synapse. Postsynaptic potentials cannot travel far in a neuron. After a few millimeters at most, the ions leak back across the membrane and the signal is lost. However, postsynaptic potentials travel far enough to reach the cell body, where they determine whether or not an action potential will be produced.
So as you can see, while those didn't actually go through the specific the specific 5 steps that entail neural impulses, it does give you a broad overview about how these things function together. And so the 5 key processes that we want to talk about here are the communication at synapses, which include the synthesis and storage, release, the binding, the inactivation or removal, and the reuptake of neurotransmitters. And the chapter talks a little bit about these different things, but as the illustration shows, synthesis and storage of the neurotransmitter molecules in the synaptic vesicles occur pretty much at the base, as we can see here. The release of neurotransmitter molecules into the synaptic cleft we can see happening at the bottom of the diagram. Then we have the binding of the neurotransmitters at receptor sites, which we were just talking about earlier. The inactivation by enzymes or removal, drifting away of neurotransmitters as they travel. And then the reuptake of neurotransmitters sponged up by the presynaptic neuron. So these are really important when we're looking at things specifically, if you want to think about it in terms of pain, okay, or pleasure, or the ways in which drug addiction occurs. These are all functions that neurotransmitters and neural impulses really explain very well. So when we're trying to explain, for instance, drug addiction, we can look at the communication that's occurring in the brain and how drug addiction impacts that exchange. So as it says, the effects of many phenomena, such as pain, drug use, and some diseases, can be explained in terms of how they alter one or more of these processes, usually at synapses releasing a specific neurotransmitter. So next we look at the postsynaptic potential. So basically what the postsynaptic potential is looking at is the voltage change at the receptor site. So that electrical exchange that's going on, what precisely is occurring there? And that is what the postsynaptic potential is looking at. So it's not all or none. There's some activity going on there. It's not one or the other. It changes the probability of postsynaptic neuron firing. Positive voltage shift equates to PSP, and the negative voltage shift looks at inhibitory PSP. So now that we looked at some of the processes that underlie this process, we also want to look at how the brain receives signals, or how different parts of the brain receive signals. Postsynaptic potential, as was on the previous slide, is a voltage change at the receptor site on the postsynaptic cell membrane. It's graded or varied in size. There's an increase or decrease the probability of a neural impulse in the receiving cell, and excitatory PSPs increase likelihood of firing, and inhibitory PSPs decrease likelihood of firing. Okay, so looking at the integration of these signals. So requires integration of signals. PSPs add up and balance out. So again, those chemical charges of electricity do end up balancing out. There is a balance between, as we just said, IPSPs and ESPSPs. Neural networks also look at patterns of neural activity. Okay, what are the exchanges that are going on in the brain at a specific point in time, and what are the different elements that are impacting that process? The interconnected neurons that fire together or sequentially, and synaptic connections, elimination and creation, and synaptic pruning. Okay, so those are all different types of signals that are integrated during this process that we need to focus on when we're exploring the activity that's going on in the brain at some point in time. So just to delve in a little bit more about neurotransmitters, we're going to watch a clip on the nervous system. So we're going to go ahead and watch that and then come back.
We know a little bit now about the communication lines between neurons. that they are powered with electricity. We also know that the messages neurons exchange are chemical molecules trapped inside these holding tanks until an electrical signal frees them. The molecules that move from one neuron to the next are called neurotransmitters. In the last decade, we detected transmitters that carry messages about pain and pleasure, about sadness and joy, about stress and relaxation. No one knows how many of these molecules of mood reside in the brain. There could be hundreds. So far, fewer than 50 have been identified. Serotonin is one of them. It's the brain's workhorse molecule, playing numerous roles, not the least of which is to inhibit our violent tendencies. Noradrenaline plays a Paul Revere kind of role. It sounds the alarm about threats from the outside world and prepares us for fight or flight. Substance P is a molecule that carries messages about pain. In a sense, Substance P is what prompts us to cry, ouch. Conversely, our brains produce painkillers, called endogenous morphines or endorphins. Neurotransmitters play a critical role in normal functioning, and yet it's easy to disrupt our chemical equilibrium. If electrical impulses go awry in the brain, the chemical transactions that are so dependent on them also go haywire. Mood disorders and mental illness can result. Change the level of a single neurotransmitter, and it could upset the chemical balance of the entire brain.
All right, so as we saw in the clip, the nervous system relies on neurotransmitters as chemical couriers to communicate information between neurons. So they're essentially messengers, sending different impulses to different parts of the brain. Specific neurotransmitters work at specific synapses. So lock and key mechanism. So looking at what neurotransmitters, looking at if they're a key, are able to unlock certain functions of the brain, meaning we can't use neurons for just any specific impulse. They have to be specific. So first, we look at an agonist, which mimics a neurotransmitter's action. We have an antagonist, opposes the action of a neurotransmitter. And there's 9 established neurotransmitters, 40 additional chemicals that function at least part-time as neurotransmitters, and a few recently discovered novel neurotransmitters. So there's a broad spectrum of different kinds of neurotransmitters, all serving a different function in the brain that looks at different abilities. Next, we look at how neurotransmitters and behavior are interlinked. And I'm not going to go through these one by one, but essentially what we want to know for how it impacts behavior is looking at how movement is controlled, how regulation of everyday behavior is controlled by the brain. And there's a variety of different things, and I'm guessing these are terms that you've heard before in other classes or in your experience studying biology or another discipline. But we have dopamine, which looks at voluntary movement of muscles, serotonin, which controls sleep, wakefulness, eating, aggressive behavior, depression, having a lot to do with the emotional reactivity and emotional regulation of individuals. And those are things, again, that we can't necessarily control all the time. we may seek out medication. For instance, depression has become a huge issue in our society, and so one of the ways to explain this emergence is looking at serotonin levels, and that's one thing that doctors and therapists are really trying to target, is how instead of just the emotional reaction, rather looking at what's going on in the brain that's causing those specific levels of serotonin to change. Okay, and then we have endorphins, which looks at endogenous morphine. Okay, and endorphins are the hormones, for instance, if you cry. Okay, there's a chemical reaction in the brain that occurs, and each time we cry, it releases endorphins to make us happier. Okay, so that also can be impacted by certain substances, drugs, for instance. So ecstasy, that's one of the things that ecstasy does. As soon as you take an ecstasy pill, it automatically triggers the function in the brain to release an abundance of endorphins. Okay, so that's what's happening when scientists look at ecstasy. So just a bit about the organization of the nervous system. So we have the central nervous system, which is the brain and spinal cord. We have the peripheral nervous system, which is the nerves that lie outside the central nervous system. And then we have the autonomic nervous system, which controls the automatic involuntary functions. The brain is traditionally divided into 3 regions, and we'll go into more depth. The hindbrain, the midbrain, and the forebrain. The reticular formation runs through both the midbrain and the hindbrain on its way up and down the brainstem. These and other parts of the brain are discussed kind of more in detail further in the chapter. The peripheral nervous system is made up of the somatic nervous system, which controls the voluntary muscles and sensory receptors, and the automatic nervous system, which controls the involuntary activities of smooth muscles, body, blood vessels, and glands. The central nervous system kind of delves a little bit more deeper into the system and consists of the brain and spinal cord. So the peripheral nervous system consists of the remaining nerves that fan out throughout the body. It's divided up into the somatic nervous system, which is shown in red in the next diagram, and the automatic nervous system, which is shown in blue. That can also be found in your book. And then finally we have the autonomic nervous system, which is composed of the nerves that connect to the heart, blood vessels, smooth muscles, and glands. It's divided into the sympathetic division, which mobilizes bodily resources in times of need. So it might be a flight or fight response. And the parasympathetic division, which conserves bodily resources. Some of the key functions controlled by each division of the autonomic nervous system are summarized later in some of the diagrams. So one of the things that's important to look at is how exactly do we study the brain and the behavior that results from specific chemical or neural impulses or brain activity. And there's a variety of different ways in which scientists have gone about studying these. So one is the lesion studies, which are important to look at. Again, these are summarized in the chapter. Brain imaging is obviously a very popular one, and a variety of others. But here what I've put down is a small representation or illustration looking at how this was studied using rats. Okay, so Olds and Milner were using an apparatus like depicted in the diagram when they discovered that self-stimulation centers or pleasure centers in the brain of a rat. In the setup, the wire delivering electrical stimulation is suspended from above so that the rat can freely can move freely about the box, and when the rat presses the lever, it earns brief electrical stimulation that is sent to a specific spot on the rat's brain where the electrode has been implanted. Okay, so again, that's looking at how exactly is an individual experiencing pain or pleasure, and what do we do in order to gain that specific experience using the brain as an explanation. Another way that we can look at the brain is obviously brain imaging, looking at computerized tomography, and then positron emission tomography, and then magnetic resonance imaging. And essentially what that's doing, and this is how a lot of studies have been done, and I think it was briefly talked about in one of the clips, but for instance, it might be looking at the different hemispheres. If we inject an individual with a certain fluid, and then we want to see what exactly the function of the brain. Let's say I want to study when someone is thinking about abstract ideas, what part of the brain is functioning during that part of the study. Then I might inject them with a fluid that's able to go into their brain, and I'm able to look at the activity in the brain. There's a variety of different imaging techniques that go along with this, but that's merely one of them. So we talked a little bit about this already, looking at the three main areas of the brain. And really, these subdivisions actually make more sense for the brains of other animals than for humans. In humans, for instance, the forebrain has become so large, it makes the other two divisions look trivial. However, the hindbrain and midbrain aren't trivial. They control such vital functions as breathing, walking, and maintaining balance. So those are things that we don't want to necessarily throw out as being trivial. The hindbrain houses the medulla, pons, and cerebellum. The midbrain holds the dopaminergic projections, reticular formation, and then the forebrain houses the thalamus, hypothalamus, limbic systems, cerebrum, and cerebral cortex. The cerebral hemispheres, two specialized halves connected by the corpus callosum, and then we'll be looking at that in just a second, looking at how exactly the brain is split. And so I'm guessing you've probably heard it, maybe not in academics, but looking at someone who is more artistic, what half of the brain controls that, or someone who is more oriented towards maybe test taking or logic, okay? So those are housed in different parts of the brain, and that's been studied looking at how exactly do we understand these different parts of the brain and how they function in terms of traits that individuals have. There's four lobes: the occipital, which controls vision; the parietal, which is looking at sensory behaviors; temporal, auditory; and then frontal, which looks at the movement and the executive control systems. Okay, so here we also have a diagram that I want to point to. The cross-section of the brain highlights key structures and some of their principal functions. As you read about the functions of a brain structure, such as the corpus callosum, you may find it helpful to visualize it. So that's kind of why I put it there. And it's on the next diagram as well. So moving on, the cerebral cortex consists of right and left halves called cerebral hemispheres. Again, the diagram provides a view of the right hemisphere, and then each cerebral hemisphere is divided in the four lobes that we just discussed. The different lobes are important to know exactly how they function together. Each lobe has areas that handle particular functions, such as the visual processing. The functions of the prefrontal cortex are something of a mystery, but they appear to include working memory and relational reasoning. So again, there's still a lot of areas that we need to consider looking at the brain that are explored, but we don't necessarily know what exactly is happening or what specific functions are housed in certain parts of the brain. That hasn't been fully identified, and those studies are still being conducted. Okay, so this PowerPoint slide looks at the hemispheres of the brain, which we briefly mentioned earlier. I've attached a small clip, so we're going to go ahead and watch that and look at how these hemispheres of the brain interact with one another and how they're different.
Six, 7, 8, 9, 10.
In almost all of us, language is processed in the left hemisphere.
This is okay. Show me how you can wiggle your fingers. Keep on wiggling.
In this test, in which the patient feels little discomfort, Jun Wada of the University of British Columbia first injects a dye opaque to X-rays into the artery supplying blood to the left hemisphere. This shows the route an anesthetic will follow when it is injected during the test. A similar series of X-rays, seen from above, shows how the dye, and hence the anesthetic, will flow only into the left hemisphere. It is vital the surgeon confirm where speech is located in each patient, so that an intended operation to alleviate epilepsy can be safely performed. The test also shows that the left hemisphere controls the right hand.
Start wiggling. Now I'd like you to count slow and loud to 10 and back to 1.
1,
Okay? Don't stop counting.
2, 3, 4, 5, 6, 7, 8.
The left hand remains upright, controlled by the still conscious right hemisphere.
Carry on.
Carry on now. Okay, carry on counting for us.
Speech gradually returns as the anesthetic clears the left hemisphere.
1,
Okay, you did very well.
What we are trying to do here is to find out which side of the brain, which side these patients have speech function. And these knowledges are very important for us when we are contemplating of operating these patients.
Although 95% of the population has speech in the left hemisphere, there are exceptions. And for this reason, it is necessary to map the geography of each patient's brain to avoid interfering during surgery with such vital functions as speech and short-term memory. In a second test, a week later, the right hemisphere is anesthetized.
Matthew, could you start wiggling your fingers? Keep on wiggling, eh? And look straight ahead. You don't have to look at me. Look straight ahead. Okay? Now start counting loud and slow.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 10, 9, 8,
The conscious left hemisphere has retained speech.
5, 4,
Great.
3,
Okay, could you show the object?
Okay. Tell me what that is.
last bubble.
Right side, make a fist.
But the anesthetic in the right hemisphere causes disoriented movements of the opposing hand.
Not at all. Show me how you can wiggle your fingers. What number?
So as we saw, each hemisphere is specialized for handling certain cognitive tasks better than others. So, for For instance, the left hemisphere, as this slide points out, it looks at verbal processing, language, speech, reading, writing. Those different functions are housed in that part of the brain, so the left hemisphere of the brain. The right hemisphere, on the other hand, looks at nonverbal processing, spatial, musical, visual recognition, kind of the more artistic part of the brain that we would think of. And as we said earlier, the cerebral hemispheres and the corpus callosum, that is what connects the two hemispheres. So be sure that you know that. In this drawing, the cerebral hemispheres have been pulled apart to reveal the corpus callosum. This band of fibers is the communication bridge between the right and left halves of the brain. The view of the left hemisphere highlights the location of two centers for language processing in the brain, Broca's area, which is involved in speech production, and Wernicke's area, which is involved in language comprehension and development. So now we move on to look at the endocrine system. And the endocrine system essentially looks at the different hormones that our brain is producing and how those hormones affect our body. So first off, hormones, as probably you are already aware, are chemical messengers in the bloodstream, okay? They serve different functions and have a variety of different uses within the body. The endocrine glands controlled by the nervous system are controlled by the nervous system through the hypothalamus, which is part of the brain that we went over earlier. I have a small graphic on the screen pinpointing where the hypothalamus is with regards to the other parts of the brain. And then we have the pituitary, which is called the master gland, and then gonads, which sex hormones, okay? So this diagram really points at the specific parts of the endocrine system, and we'll kind of go into that later. Okay, next we want to talk about heredity and behavior. So the basic principles of genetics are laid out in this chapter. And the first thing they talk about is chromosomes, which are strands of DNA carrying genetic information. These are the things that, for instance, determine the sex of a child, okay? Human cells each contain 46 chromosomes in pairs, so sex cells, 23 single. Each chromosome, there's thousands of genes also in pairs, okay? So it's kind of a transcript, if you will, of a record of our genes. Each parent contributes 23 chromosome pairs, which can be scrambled in over 8 million ways. Okay, so that's why there's such diversity in the world. I mean, these chromosomes have such variance in what they're going to produce that that's an important facet of the heredity and behavior. More distant relatives share smaller portions of genes, which isn't surprising, right? Our immediate family share more genes than relatives that might be a little bit more distant. And then identical twins share 100% of their genes, okay? And so this is really important when we're looking at how exactly we study genes and chromosomes and heredity. And one of the ways in which we do it, and we talk a little bit later about this when we go into development, are through family studies, twin studies, and adoption studies. Research on the genetic bases of behavior takes advantage of the different degrees of genetic relatedness between various types of relatives. If heredity influences a trait, relatives who share more genes should be more similar with regard to that trait than are more distant relatives who share fewer genes. So comparisons involving various degrees of biological relationships will come up fairly frequently in later chapters. And lastly, most human characteristics are polygenic or influenced by more than one gene. So one gene doesn't dictate a specific behavior, rather there is an interaction between those genes. They're not mutually exclusive. So those are interrelated. And as we see on the diagram over here, the degree of genetic overlap between unrelated 0%, second cousins, first cousins, and so on and so forth, until we get to identical twins, which is 100%. Okay? Okay, and lastly, we want to look at evolutionary psychology. So again, we went over that a little bit in chapter 1, but it's based on Darwin's ideas of natural selection and reproductive success as a key. So specific brain functions that allowed us to adapt in specific environments were key when we're talking about the development of the brain. So for instance, if we want to talk about emotions, okay, a lot of emotions are actually socially constructed rather than biological parts of the brain, where I think there's 6 primary emotions housed in the frontal part of the brain, or the back of the brain. And then as we develop and we socialize, our brain actually changes to adapt to our social surroundings, interestingly. Adaptations, behavioral as well as physical, okay? So we not only adapt physically as a species, but also our behaviors, whether they be social behaviors, instinctual behaviors, they're adapted in order to survive in specific environments. The example that was used in the chapter, for instance, is the taste preference, okay? Looking at the different types of taste. And we talked a lot about this in chapter 2, or in chapter 1 rather, looking at how individuals' taste is developed. So that concludes the brain and exactly what is occurring in the brain during our different processes. If you have any questions, please feel free to email me, and I will see you next week.